I can tell you a little bit about the origins of McSAS3. It was a program that was very much developed out of a need, or rather a frustration that I had with the classical fitting methods. And this was quite a while ago already. This was during my time at the Spring 8 synchrotron in Japan. It's a fascinating synchrotron. I met lots of interesting people. And over there we did experiments on hydrothermal synthesis, right? So high temperature, high pressure experiments. So we were at the small angle scattering beamline over there, where we had installed this setup so we can measure through a sapphire capillary and observe the formation of, for example, zirconia particles under hydrothermal conditions. The problem was that the data that we got out of that was very difficult to analyze, in that I could not find a classical fitting model that could fit all of the stages of the synthesis. I wanted to be able to fit the initial stages of particle formation as well as the growth of the particles. But none of what I tried actually worked. So I started experimenting with alternative methods for analysis. And so McSAS was developed. I was quite surprised that it worked myself, but it's a very pragmatic, sort of brute force way of getting some structural insight into what's going on in your sample. Now, let me show you a couple of examples of what we've been using McSAS for. Now, there's like a couple of hundred papers that cite McSAS, so it's been used for a very wide range of stuff. A couple of highlights I can show here. For example, one of the examples that we have involves that of ceramic nanoparticle composites. This is a project by a colleague of mine who is studying who's trying to structure purely ceramic compounds in 3D. And for that, she needs these ceramic composites. What she wanted to know was whether the particles inside the polymer matrix were distributed well, or whether they were actually clumped together. And so we did a series of measurements on the current instrument on a range of composites with an increasing amount of volume fraction. So you can see over here, it's actually a really interesting data set, because as you increase the volume fraction, you start to see the structure factor coming in, right? The interparticle interference coming in the dark green curve. And then that sort of bump shifts to higher and higher angles until it meets up with the first sort of shoulder of our form factor. What you can also see in this is that we measure all the way into the wide angle range. Interestingly, if you do your measurements on absolute scale, the amount of wide angle signal that you see, so these wide angle diffraction peaks, are actually proportional to the volume fraction of crystalline material that you have in your sample. So you now have two ways of figuring out the volume fraction of component in your sample. One is to figure out the amount of crystalline material you have in your sample by looking at the height of these diffraction peaks. And the second thing you can do is to look at your small angle scattering signal on absolute scale, which will also tell you what kind of volume fraction you have over there. And since this is a dense data set, you might now be able to play around with structure factors and find out how far you can go and what kind of analyses you can do on this data to get more information out. So we haven't fully analyzed this data set, but we do have a couple of data sets figured out. So one of them is, of course, the low concentration version. This one is shown over here. So in orange, you can see the size distribution. Do pay attention that the size distribution horizontal axis has been inverted to match the information in the scattering pattern. But the fit, of course, is very nice, as is usually the case with McSAS. And it matches up very nicely with the information that we get from electron microscopy on the right. Now, dilute samples are all good and fine, but what about concentrated samples? Well, you can now also introduce a volume fraction. You can now also introduce a structure factor in your model, so that you can analyze dense samples. And this can be very helpful, for example, here in the case of the composite with a high volume fraction of filler, where you see this clear structure factor peak. You can still get size information out. It can also help you when analyzing powders. Now, one of the current things that we do a lot is to measure metal organic framework powders using our instrument. And since these are powders, the particles are actually very close together, and we need a structure factor to be able to analyze the data. So in introducing a structure factor is not a big problem. The main issue with that is that you need to know the volume fraction, the volume fraction for your structure factor. And this can be a little bit tricky. It is something that we hope to be able to figure out with this dense composite data set. It's been on my to-do list for the last couple of years. But it's certainly worth exploring. But at least now you know that, if you have dense systems or something, you can still use McSAS and get size distributions out. One of the earlier examples for McSAS involves a study on formation of quantum dots. So this was a study by Benjamin Abecassis and colleagues from France, and they wanted to know by what mechanism these quantum dots form. So they did what you usually do. They set up an in situ experiment at a synchrotron by having a lab scale reaction right next to your instrument, attaching a tube to a pump, so you can pump out a little bit of the reaction liquid, pump it through your measurement cell, and then pump the remainder back into your reaction flask. And this is actually the best way of doing in situ experiments, as opposed to trying to do reactions in your capillary. If you try to do reactions in your capillary itself without actually moving the liquid, the kinetics will be very far off, or could be very far off, from the normal lab scale reactions that you do. So try and do things on the lab scale and then pump a little bit of the reaction liquid through if you can. Much more consistent results, much lower chance of actually affecting the kinetics with your X-ray beam. And of course, in this case, if you want to start a reaction, you have to add some heat to the reaction vessel, which is exactly what they did. So after some initial attempts, just like us, after some initial attempts with classical models, they ended up using McSAS because of the complexity of the particle formation. So you see on the very left-hand side, you see all of the scattering patterns in color and the fits in black. And right next to that, you see the distributions, the size distributions. And you see what's happening is that you actually get one distribution that splits into two. This is why classical models don't work. For this particular example, it splits into two, and then one of the populations decreases while the other one increases in amount as well as in size. And when you then convert this to key parameters such as yield, polydispersity, and the mean radius, that allowed the researchers to eliminate 2 out of 3 potential pathways and thereby verify one particular pathway with a rate-limiting step of the selenide precursor for the formation for these quantum dots. So that's a couple of examples slightly more in depth. We also have other examples for which we've used McSAS. You can use them very well for porous carbons, all kinds of patterns with some humps and some bumps, smoothly varying scattering patterns, as you see in the top left-hand side. And you can get all kinds of size distributions out. And you see that it's nice that you now get size distributions out. On the other hand, you now have a different problem, right? You now need to convert the information in these size distributions to something that you can actually use and compare. So we basically shifted the problem from the analysis of the scattering pattern to the analysis of the size distribution. Another example, which is quite neat, I think, is an example where we adapted McSAS for the analysis of objects for which we don't have form factors. So we don't have a form factor, or not an analytical form factor at least, for a rounded cube, which we needed for this particular sample. So we used a simulation. Well, we generated an STL file of a rounded cube, as you can see on the top right hand side. We calculated how that would scatter, so I have a different program for that. And that gave us the scattering of a 1 nanometer cube. So we used that as a base model for McSAS, and McSAS then was able to to scale that and fit it to the data, so that you could get a size distribution out of the data. Now these particles, they came from the PTB, and they always make wonderful monodisperse or very low dispersity samples, very far away from many of the industrial materials that come across our desk. But nonetheless, very interesting, and perhaps an interesting application if you have samples that don't have a specific form factor predefined. Lastly, one of the oldest examples is that of the growth of cylindrical precipitates in magnesium-zinc alloys. So you might not know, but metallurgy was one of the first applications of small angle scattering in the very early 1900s. Dubai and Guinea and Preston, they were all sort of X-ray specialists. And for metallurgy, it's quite interesting because the precipitates that form, so you have these sort of particles that form inside of your metal alloy, and they very much affect the mechanical properties of your alloy. So it's very important to understand how they grow, and if you're doing particular temperature cycling or heat treatment, it's very important to know how this particle growth is affected by that heat treatment, how you may be able to tune it. So we have these cylindrical precipitates growing inside magnesium-zinc alloys. So we use this a model with cylinders in our McSAS analysis. The data and the fits are shown on the right-hand side. As I mentioned, this is very old data, so it's from a very old instrument. That's why the range is so short. Nonetheless, we got results out of it, which we could compare to electron microscopy. So you see the small angle scattering distributions in red, and the electron microscopy distributions in the lower half of each graph in green and blue for the samples for which they were measured. Now, realize that for the small angle scattering measurements, we're averaging over billions of particles, whereas for the electron microscopy data, these were painstakingly measured, hand measured, artisanal, I would say, for about 500 precipitates for each sample. So it's a very tedious, painstaking work with electron microscopy. But this also shows one of the advantages, right? If you combine electron microscopy, because we need to know the shape, right? If you combine electron microscopy with small angle scattering, that combination is super powerful. The one gives you the shape of your precipitates, and the other one gives you the size distributions and the volume fractions with very low effort.